So now that we have a broad overview of what happens during fertilization, let's put this into context of a specific species model organism that we can see direct events of fertilization in. So what we're going to be doing now is looking at a model organism that is of great use to developmental studies, and that is the sea urchin. So we'll look at sea urchin fertilization. In the next couple of flowcharts, we'll entitle this first one Sea Urchin Fertilization 1. And remember, the sea urchin is a part of the Echinodermata phylum. And here, so of course, we're still looking at an animal, but we're looking at a rather simple animal. This is an animal that, of course, lives in the water. And this is going to be critical for the overall sort of understanding of how it develops and how it specifically fertilizes. As we go through this idea of sea urchin fertilization, a good summary figure of everything that we'll talk about is figure 47.13. That does a good job of looking at and visualizing everything that we're going to say from this point forward. So first of all, why a sea urchin? Of all things in the world, why would you care, or why does it even matter that a sea urchin fertilization is necessary to understand anything about development? There are some major advantages to using something like a sea urchin. First of all, sea urchins are quite readily available. In other words, these are not endangered animals. These are not animals that are rare. They are very sort of prominent. They are successful in their environments, and thus we can take them and view them very easily without disrupting anything in terms of ecosystem consequences. So they're readily available. They're also very easy to work with. They're very easy to work with. Now, what does that mean? That simply means that they're easy to maintain. They're not hard to feed. They're not expensive to feed. They're not hard to sort of reproduce. They are very sort of manipulative. They can be manipulated, let's say. They can easily be sort of contoured and adjusted to what a scientist needs to understand about fertilization in this situation. So they're easy to work with. In addition, this is another sort of idea of why they're easy to work with, sort of in terms of context. They produce lots of gametes. So when they produce gametes, they produce lots of sperm and lots of egg. That means that there's a lot of chances for us to view fertilization events. So that's a very good advantage if you're trying to study fertilization in an organism. In addition, I think this is the biggest advantage, the one that you have to remember, is that sea urchins are external fertilizers. They are external fertilizers. Now, why is that? They live in the water. The water is a medium in which sperm can float and swim. Water is a medium in which egg can float and swim. And because we have an external fertilization of egg and sperm swimming and meeting towards each other, we can then, as far as our advantage is concerned, we can view all steps of fertilization externally. We don't even have to go on the inside. We don't even have to do this in vivo, let's say, within an organism as it's living. We can see all this happening on the outside. Thus, these are major advantages to using something as seemingly random as a sea urchin, and thus it makes sense to use this model organism. So, let's take a look at the sea urchin and how it helps us understand fertilization overall. So remember when we talked about the egg? The egg has many coverings and it depends on the species. That's what we're going to be looking at first, because that's an initial understanding we have to have in order to understand what the sperm has to do to eventually meet on the inside of the egg's nucleus and fuse with the nucleus, as we'll see. So it's important to first look at the egg structure of a sea urchin, okay? And as we're doing this egg structure, what we're going to be doing is going from inside to outside. We're going to go from the innermost layer of the egg to the outermost layer of the egg. So let's label that out. So what we first have is the innermost. The innermost layer, the innermost structure you want to understand is the plasma membrane. So this is where sperm essentially has to get to. Absolutely has to get to. But it has to first pass, or it has to pass before this, something, the middle layer, which is known as the vitelline layer. Okay, so VITELLINE. VL layer, VL from this point forward. This is just considered the thin layer of the egg. So this is a middle layer, and then the outermost layer, this is the first layer that the sperm has to sort of go through. It's a thick layer. It's called the jelly coat. Not the jelly coat, the jelly coat. JC from this point forward. Now, like I said, this is a thick layer. Why do you think it's thick on the outside? Well, that's because it's very protective of the inner very less stable structures on the inside. Thus, it's important to put a nice thick structure, stable and strong on the outside. And most importantly, the jelly coat also contains these very important things that you've seen many times before called glycoproteins. What have we been constantly referring to glycoproteins as thus far? We've been constantly saying glycoproteins are always involved in recognition. And what do you think is going to be the recognition event here? Sperm receptors recognizing egg receptors and causing consequences, causing eventual fertilization events to start and begin and then conclude, of course. So that's our egg structure. Sperm has to meet the jelly coat, digest through it, get through the vitelline layer, and then eventually deposit its nucleus into the plasma membrane and into the internal cytoplasm of the egg. So that's what we're going to be doing. So that's our sort of overview of what the sperm has to do. Let's see the specific steps associated with that. So now let's go over the steps broadly. All these steps are highlighted nicely in this figure, so be sure to look at this figure as we're going through this. So step 1, what do we have to do first? What's the first layer that we have to encounter? The jelly coat. But specifically, remember, the sea urchins are external fertilizers. So sperm has to somehow meet up with egg in this huge water environment that they live in. How is that possibly going to happen randomly? How is that possibly going to happen? Well, it's not going to be quite random as we think at least. What's going to happen is, amazingly, the jelly coat of eggs that are also released into the water, the specific structure known as the jelly coat, the outermost layer, is going to do the following to ensure that sperm meets egg. Jelly coat of eggs releases chemicals into the water. Releases chemicals into water. Chemicals are signals. Signals for something right now. Who do you think is going to be attracted to these chemicals that the egg is releasing? Of course, that's going to be the sperm. This attracts the sperm. These chemicals amazingly attracts sperm via a specific sort of mechanism called chemo, referring to the chemicals that are released, taxis. Taxis means movement. So movement via a chemical gradient that is coming from the eggs. So the sperm moves towards, it taxis towards the chemicals. And how does it do that? Why does it do that? Because the jelly coat told it to. The jelly coat of the egg attracted the sperm. So that's sort of how we meet sperm and egg. Okay, good. Now, once they've met, I should say, what's going to happen? So we're going to have sperm plus JC for jelly coat are going to meet, and this is going to result in something we've talked about before, known as the acrosomal reaction. Remember sperm structure. The sperm has a head, a midtail, a midpiece, and also a tail. The head of the sperm has a region known as the acrosome. Okay? And this is the same for sea urchins as it is for human sperm. The acrosome, acro just means tip and some means the body. The structure is going to be, of course, let's reiterate, at the head of the sperm. The very tip of the sperm head will contain this acrosome structure. What does it do? If you remember, it releases those very important hydrolytic, these breakdown enzymes that are very good at breaking down structures. And what do you think this is going to break down? Sperm meets jelly coat. Jelly coat is a thick layer. So of course, in order to digest through the thick layer, you release the hydrolytic enzymes from the acrosome so that you can have this digestion. So we'll say that this event partially digests the JC, the jelly coat. And this is going to be all because the sperm has recognized the specific jelly coat of the same species and thus triggered the acrosomal reaction. All of this is going to then culminate and sort of create something known as the acrosomal process. The acrosomal process, which is a structure, forms. Now, what does this structure entail? The acrosomal process is the sort of name for the lock and key mechanism that we've sort of highlighted and sort of mentioned in our initial overview of fertilization. When you have the lock and key mechanism successfully occur, you've created an acrosomal process in which you have the sperm head, that's the tip, that's the structure that recognizes, that structure produces something known as an actin filament So this is just a protein filament, a protein sort of highway structure. I like to think of this as a road, an actin filament structure that penetrates the coat. So this is sort of a pathway. Remember how we had to make that pollen tube in plants way back when? I like to think of this as an analogous version of that. Remember you had those two cells, you had the pollen tube, and you also had to then release and deposit the actual sperm cell? Here we're creating sort of a structure that goes straight through that thick jelly coat so that you can then deposit the sperm's nucleus into and eventually through the plasma membrane. And that's what we're doing here essentially after the lock and key has been fit. So now we then state that the sperm will have specific protein molecules that are going to sort of construct this. Sperm protein molecules bind to specific plasma membrane receptors of whom? The plasma membrane receptors are specifically of the egg. You have this binding, you have this double sort of lock and key event. Why? Why do you have this check and balance that's going to ensure that sperm locks and its key, the lock and key of sperm and egg fits perfectly together? All of this is just a very sort of broad way that we can sort of label all of this as the following. All of this sort of repetitiveness, it seems like, is important because it ensures intra-specific. Now, please note, I said intra-specific fertilization. Before, we stated that we wanted to prevent interspecific fertilization. Now I'm telling you I want to ensure intraspecific fertilization. Intraspecific means within the same species, not between different species, intraspecific, within the same species fertilization. How do you do it? Make sure lock and key fit, make sure sperm can easily deposit its sort of nucleus and genome into the egg through this coat digestion that we're doing, and then eventually the binding and recognition process that we've highlighted before, all a part of the acrosomal reaction that we see. So those are our initial steps. We'll continue and conclude the sea urchin fertilization steps and story in the next video.